In this script, we will delve into the intricacies of the cAMP signaling pathway, a fundamental process in cellular signaling. We will explore various topics including the basic principles of the pathway, the roles of G protein-coupled receptors, GPCRs, adenylyl cyclase activation and regulation, the functions and mechanisms of cAMP-dependent protein kinases, pKas, diseases associated with dysregulated cAMP signaling, the hepatic pathway and its components, calcium release and protein kinase C, PKC, activation, the physiological significance of the hepatic signaling pathway, an introduction to MAP kinases, and their involvement in intracellular signaling. We will conclude with a summary of the key concepts discussed. The cAMP signaling pathway is a crucial mechanism used by cells to relay extracellular signals to intracellular responses. It involves the conversion of adenosine triphosphate, ATP, to cyclic adenosine monophosphate, cAMP, by the enzyme adenylyl cyclase. cAMP acts as a second messenger, transmitting the signal from cell surface receptors to effector molecules within the cell. This pathway plays a pivotal role in numerous physiological processes such as cell growth, metabolism, and gene expression. GPCRs are a family of cell surface receptors that mediate responses to a wide array of extracellular signals. They play a crucial role in initiating the cAMP signaling pathway. Upon binding to specific ligands, GPCRs undergo conformational changes that activate heterotrimeric G proteins. These G proteins, in turn, regulate the activity of adenylyl cyclase, leading to cAMP production. Adenylyl cyclase is an enzyme responsible for synthesizing cAMP. It is regulated by various factors, including G proteins, calcium ions, and phosphorylation. When activated by G proteins, specific isoforms of adenylyl cyclase catalyze the conversion of ATP to cAMP. Regulation of adenylyl cyclase activity is crucial for maintaining the appropriate levels of cAMP and ensuring precise signaling responses. cAMP-dependent protein kinases, or pKas, are key effectors in the cAMP signaling pathway. They are composed of regulatory and catalytic subunits. Binding of cAMP to the regulatory subunits induces a conformational change, leading to the release and activation of the catalytic subunits. Activated pKas phosphorylate various target proteins, modulating their activity and initiating downstream signaling cascades. Dysregulation of the cAMP signaling pathway has been implicated in the pathogenesis of several diseases. For instance, mutations in GPCRs or adenylyl cyclase isoforms can lead to aberrant cAMP signaling, contributing to disorders such as endocrine tumors, cardiac arrhythmias, and cystic fibrosis. Understanding the intricate details of the cAMP pathway is crucial for developing targeted therapeutic strategies for these conditions. The inositol 1,4,5-trisphosphate, IP3, and diacylglycerol, DAG, pathway, commonly referred to as the hepatic pathway, is an alternate signaling mechanism utilized by cells. It involves the activation of phospholipase C, PLC, and the subsequent generation of IP3 and DAG as second messengers. IP3 acts on IP3 receptors located on intracellular calcium stores, releasing calcium ions into the cytoplasm. DAG, on the other hand, activates protein kinase C, PKC. Phospholipase C, PLC, is a key enzyme responsible for initiating the hepatic pathway. PLC is activated by various extracellular signals, such as GPCR-mediated activation or receptor tyrosine kinase signaling. Upon activation, PLC hydrolyzes phosphatidylinositol 4,5-bisphosphate, PIP2, generating IP3 and DAG. IP3 diffuses through the cytoplasm, binding to IP3 receptors on the endoplasmic reticulum, leading to calcium release. DAG remains in the membrane, activating PKC. IP3 plays a pivotal role in calcium release from intracellular stores. Binding of IP3 to IP3 receptors on the endoplasmic reticulum triggers a conformational change, opening calcium channels. This allows calcium ions to flow from the endoplasmic reticulum into the cytoplasm, increasing the cytosolic calcium concentration. Calcium serves as a versatile second messenger, regulating various cellular processes, including neurotransmission, muscle contraction, and gene expression. DAG, a lipid second messenger, plays a critical role in the activation of protein kinase C, PKC. DAG recruits PKC to the plasma membrane, allowing its interaction with calcium ions and phospholipids. These interactions induce conformational changes in PKC, activating its catalytic domain. Activated PKC phosphorylates a myriad of protein substrates, modulating their activity and initiating downstream signaling events. The hepatic signaling pathway plays a vital role in cellular physiology and pathogenesis. It is involved in a wide range of cellular processes, including cell growth, differentiation, apoptosis, and immune response regulation. Dysregulation of this pathway has been associated with several diseases, including cancer, neurological disorders, and immune system dysfunctions. Elucidating the intricacies of PDAG signaling is crucial for understanding disease mechanisms and developing targeted therapeutics. MAP kinases, also known as mitogen-activated protein kinases, are a family of serine/threonine kinases that play a central role in intracellular signaling. They are involved in transmitting extracellular signal inputs to various intracellular targets, including transcription factors, cytoskeletal proteins, and cell cycle regulators. MAP kinases are crucial for coordinating cellular responses to diverse stimuli such as growth factors, cytokines, and cellular stressors. MAP kinase modules constitute a three-tiered signaling cascade involving MAP kinases, MAP kinase kinases, MKKs, and MAP kinase kinases, MAPEX. Upon stimulation, MAPEX activate downstream MKKs, which, in turn, activate MAP kinases. Activated MAP kinases then phosphorylate target substrates to initiate specific cellular responses. This cascade ensures signal amplification and precise transmission of extracellular cues within the cell. In conclusion, the cAMP signaling pathway and the PDAG pathway play vital roles in cellular signaling, coordinating a myriad of physiological processes and influencing disease progression. Understanding the complex mechanisms and interplay between these pathways is crucial for unraveling disease mechanisms and developing targeted therapies. Additionally, MAP kinases serve as key mediators, integrating and transmitting extracellular signals to the appropriate intracellular effectors. Future research on these signaling pathways holds great promise for further unraveling the intricacies of cellular communication and health.